Hello students, this is the 11th session of the unit 3, that is the traditional methods of food preservation. In the previous sessions, you have learned about food additives, that is the definition, classification, their functions and its uses in various process products. And now we will discuss about the fermentation, its types and the microorganisms involved in fermentation. So, what exactly is fermentation? Fermentation is a process that helps break down large organic molecules via the action of microorganisms into simpler ones. For example, yeast enzymes convert sugars and starches into alcohol, while proteins are converted to peptides or amino acids. Bennion describes fermentation as a general term which covers aerobic and anaerobic changes brought about by microorganisms and includes the production of acids and alcohols. This process also produces heat and copious amounts of carbon dioxide as byproducts. Bacteria and fungi are generally involved in fermentation. The bacteria concerned with fermentation are lactic, acetic, butyric, and other acid forming groups. The fungi concerned in fermentation are the yeasts. The microbial or enzymatic actions on food ingredients tend to ferment food, leading to desirable biochemical changes responsible for the significant modification to the food. Fermentation occurs at specific temperature conditions. Too low or too high temperatures terminate fermentation. The food items with large amount of sugar and starch easily undergo fermentation. Next, the common groups of microorganisms involved in food fermentations are bacteria, yeasts, and molds. The most important bacteria in food fermentation are Lactobacillus, Pediococcus, Streptococcus, and Enococcus, which have the ability to produce lactic acid from carbohydrates. Others are acetic acid producing bacteria such as Acetobacter species, which produces acetic acid by fermentation of fruits and vegetables, and Bacillus species which produce acetic acid from fermentation of legumes. The beneficial yeasts in terms of desirable food fermentation are from the Saccharomyces family, especially Saccharomyces cerevisiae. It is involved in bread making and alcohol in the wine fermentations. Schizosaccharomyces pombe and Schizosaccharomyces boulardii are the dominant yeasts in the production of traditional fermented beverages, especially those derived from maize and millet. Molds involved in food fermentation are Actinomyces, Aspergillus, Fusarium, Lecanicilium, Mucar, Neurospora, Penicillium, Rhizopus, and Sperendonema. Then, with regard to the types of fermentation, there are three types of fermentation. Number one, ethanol fermentation. Number second, lactic acid fermentation. And third, acetic acid fermentation. We will now move on to ethanol fermentation. Ethanol fermentation is the process in which glucose is converted to ethanol and carbon dioxide. Ethanol fermentation is also known as alcoholic fermentation that converts sugars such as glucose, fructose, and sucrose into energy, ethanol, and carbon dioxide as byproducts. One molecule of glucose is converted to two molecules of ethanol and two molecules of carbon dioxide. In the absence, perform this conversion in the absence of oxygen. Hence, alcoholic fermentation or ethanol fermentation is considered an anaerobic process. Here, glucose is broken down into pyruvate, and pyruvate in turn is converted to two molecules of ethanol and two molecules of carbon dioxide. Next, moving on to lactic acid fermentation. Lactic acid fermentation is a process by which glucose or other 6 carbon sugars, also the disaccharides such as sucrose or lactose, are converted to energy and lactic acid. Lactic acid fermentation is strictly controlled by salt concentration. Lactic acid bacteria fermentation is done by using natural microflora or lactic acid bacterial cultures. The most commercially important genus of lactic acid fermenting bacteria is Lactobacillus. Two of the most common applications of lactic acid fermentation are in the production of yogurt and sauerkraut. Sauerkraut is finely cut raw cabbage that has been fermented by various lactic acid bacteria, and these bacteria ferment the sugars in the cabbage leaves and form lactic acid. Lactic acid is naturally produced in fermented products from the sugar present in the food sample. Lactic acid bacteria is a group of gram-positive, non-spore forming cocci or bacilli which produce lactic acid as a major end product. The basic mechanism of the preservation of food is the production of acid, chiefly by lactic acid bacteria, which lowers the pH to a level at which most of the spoilage causing microorganisms cannot grow, and thus the food is preserved. Move on to acetic acid fermentation. Acetic acid bacteria. Next, we will move on to acetic acid fermentation. Acetic acid bacteria are a group of gram-negative bacteria which oxidize sugars or alcohol and produce acetic acid during fermentation. The acetic acid bacteria consists of 10 genera in the family Acetobacteriaceae. Several species of acetic acid bacteria are used in industry for production of certain foods and chemicals. Vinegar is formed when acetic acid bacteria is added to alcoholic beverages. In this process, oxidative fermentation takes place.